Good morning, everyone. My name is Trish Secca, and I'm a Senior Nutrition Program Manager here at IAFNS, the Institute for the Advancement of Food and Nutrition Science. Today, I have the honor of moderating this webinar, FDA Regulatory Review Process for Food Additives, including low and no calorie sweeteners. This webinar is being provided as part of a research study being conducted out of the University of Massachusetts on healthcare practitioners' perceptions of low and no calorie sweeteners. Following the webinar, participants for the research study will receive a link to a short post-webinar survey to complete their participation in the study. Before we get started, I just want to let attendees know how they can interact with us and ask questions. So you may see a screen like this with a orange arrow for your control panel. That's how you can collapse and expand the control panel. And in the below area, you can ask questions. Questions are only visible to staff for the webinar and not to other attendees. If you have an older version of GoToWebinar, you may see a screen like this. You can interact with us the same way and ask questions here, and we will address them at the end of the webinar during the Q&A period. Just want to remind participants that this webinar is also approved for one continuing education unit with the Commission on Dietetic Registration, and information will be provided how to access your certificate after you complete the post-webinar survey. So let's get started. Our speaker today will be Dr. Anne McGregor-Das, and joined the FDA in 2020 as a biologist in the Toxicology Review Branch within the Division of Food Ingredients in the Office of Food Additive Safety. In her role, Anne serves as a safety reviewer for generally recognized as safe GRAS notices, as well as food and color additive petitions. She currently serves as an FDA liaison to the Institute for the Advancement of Food and Nutrition Sciences on our Low and No Calorie Sweetener Committee. She's also co-chair for the Society of Toxicology and the FDA Colloquium Organizing Committee. Prior to the FDA, she worked at the Johns Hopkins School of Medicine, conducting research on genetic biomarker discovery and pancreatic cancer screening programs, and received her PhD in pathobiology from the Johns Hopkins School of Medicine in 2015. Please welcome Anne.
Thank you, Trish. Good afternoon, everyone, and thank you for attending this webinar. I'm very much looking forward to giving today's talk on the FDA regulatory review process for food additives. So the objectives of today's webinar are to describe two of the main regulatory pathways for food additives in the United States, which include our pre-market approval process for food additives, known as a food additive petition, as well as the generally recognized as safe or GRAS provision. Then I'll move on to cover an overview of the safety assessment that we use for food ingredients. And finally, I'll provide a case study of the GRAS evaluation for the sweetener Steviol glycosides. So let's dive in. Why are food ingredients added to food? Well, additives perform a variety of useful functions in foods that consumers will often take for granted. Most consumers today rely on the convenience that additives provide. So on this slide, you'll see some reasons why ingredients are added to foods. First, they are added to maintain or improve safety and freshness. Preservatives, for example, can slow down product spoilage caused by mold or bacteria, or they can control contamination that could cause foodborne illness. Antioxidants, for example, can help prevent fats and oils and foods containing them from developing off flavors. And a second reason is to help improve or maintain nutritional value. So vitamins and minerals can be added to many foods to make up for those that are lacking in a person's diet or that would otherwise be lost in the processing steps. This fortification and enrichment has helped to reduce malnutrition throughout the US and worldwide. And finally, some ingredients can improve the taste and texture and appearance of food. So for example, spices, can be added to help enhance the taste of food, and some additives help control the acidity of foods, while others will give foods the texture and consistency that consumers expect. So now that I've introduced why ingredients are added to foods, let's consider the regulatory implications of this. I'll begin with an overview of the Federal Food, Drug, and Cosmetic Act. We call this the FD&C Act, and it was passed in 1938. And in addition to providing the regulatory framework for drugs and cosmetics, it also gives the FDA the authority to regulate food and food ingredients and to oversee the safety of the food supply. And then this regulatory framework was further described in 1958. We have the Food Additives Amendment to the Act, which was passed by Congress in response to concerns about the safety of new food additives. And it is in this amendment that we get the definition for both a food additive as well as substances whose uses are concluded to be GRAS. And in addition, it establishes the standards for review and safety, as well as the formal rulemaking procedures. So the first regulatory pathway I will discuss today is the food additive petition pathway. But let's start by defining a food additive. So the 1958 Food Additives Amendment defines food additives as any substance, the intended use of which results or may reasonably be expected to result directly or indirectly in it becoming a component or otherwise affecting the characteristics of food. So this is a fairly broad definition that basically encompasses any substance that may be added directly or come into contact with food. This includes substances that may be important in, say, the processing or the manufacturing or packaging of food, which today is not the focus of this talk, but I will note that we have an office in my division that oversees the pre-market approval of these food contact substances. So there are always exceptions, and this is also the case for food additives as well. Within the regulation defining food additives, there are several exceptions that were prescribed. This includes ingredients that were approved for use by the FDA or the U.S. Department of Agriculture before the 1958 Food Additive Amendment was passed. Additionally, there are also exceptions for color additives, which have their own separate regulations. Pesticides, which are regulated by the EPA, as well as animal drugs and dietary supplements, which have their own pre-market approval requirements. So in the US, the FDA has the primary legal responsibility for determining the safe use of food additives. This means that before food additives can be marketed, a manufacturer must submit a petition where they propose a regulation outlining the conditions of use under which their food additive can be safely used. Alternatively, these manufacturers could also submit an amendment to an existing food additive regulation if they wish to amend the uses that are already approved. So what would a food manufacturer include in a food additive petition? The specific requirements are also prescribed in the Code of Federal Regulations, or CFR. Specifically, Title 21 of CFR is reserved for rules of the FDA. So you'll see on my slides nomenclature throughout that refers to the regulation and where it's found within 21 CFR. When evaluating the safety of food additives, FDA considers the identity and composition of the ingredient, the intended uses, and the amount of that would be typically consumed. And we also evaluate safety data and information on the substance to establish with a reasonable certainty that no harm would result from consumption. So I hinted at that the previous slide, what the safety standard that we use. And as previously mentioned, that safety standard language is prescribed directly in the 1958 Food Additive Amendment, where it states that safety requires proof of a reasonable certainty of no harm from consumption of the substance. And so what does this mean practically? Well, due to the inherent limitations of science, FDA cannot guarantee with absolute certainty the absence of any risk from consumption. So we evaluate the available science to determine whether there is a reasonable certainty of no harm to consumers. Importantly, if new science emerges and there's evidence to suggest that an ingredient that is already in the food supply may be unsafe, or if we have reason to believe that consumption levels have changed, FDA can either prohibit its use, or they can conduct further studies to determine if the use is still safe. Ultimately, for a food additive petition, FDA has the primary legal responsibility for determining the safe use of food additives. So this means that while the burden to establish this proof rests on the petitioner or the food manufacturer, the safety decision ultimately belongs to the FDA. So to achieve this safety standard of reasonable certainty of no harm, FDA evaluates the safe use of a food additive petition using a multidisciplinary team of scientists, and we often obtain scientific expertise for our reviews from other scientists throughout the agency as needed. I briefly mentioned the components of a food additive petition a few slides ago, and listed on this slide are the roles and responsibilities of each member of the review team. So this team typically consists of a chemist and a toxicologist, an environmental scientist, and then when needed, we can include a microbiologist or other scientists such as a pathologist or a nutritionist. And then this entire review process is managed by a regulatory review scientist. These RRSs, as we call them, are scientists as well, with knowledge and experience that contributes to the overall review process. So what does this review process look like? Well, the review team members independently review the petition and will make an initial decision about whether there is sufficient information to file. And if there is, then each team member conducts their own personal, robust review of the available data and science, along with whatever we find in the peer review literature. Typically, we often have questions for the petitioner, which we will supply to them, and they provide an amendment with responses to our questions. This process is often very iterative, with many rounds of back and forth questioning until FDA is satisfied that the data that is provided supports safety. And finally, a successful food additive petition ultimately results in a regulation in the CFR. So that was a brief overview of the food additive petition process. And again, on the left-hand side, you'll see the definition of a food additive as defined in the Act. But in defining food additives, Congress decided to include an exception for substances for which there is an expert consensus about its safety under provided intended uses. And we call this the GRAS provision, and the language is shown here in the box on the right. GRAS stands for Generally Recognized as Safe, and I'll spend the next few slides looking at the GRAS provision a little bit more closely. There are 2 points that I would like you to take away from this slide. First, the Act dictates that FDA must approve the use of food additives and issue a regulation as part of the approval process or the petition, food additive petition process that I just described. In contrast, if a substance meets the GRAS criteria, it does not require pre-market approval by the FDA. In effect, what that means is that the GRAS notification program is voluntary. Second, if you look at the language of both a food additive and a GRAS substance, you can see that Congress was very deliberate prescribing language that safety is tied to the intended use of the ingredient. It is not actually the food ingredient that is deemed safe or unsafe, but rather the intended uses. In other words, we're not saying that a particular sweetener, for example, is safe at whatever amount and in whatever foods a company wants to put it in. Instead, our conclusions are saying that the sweetener is safe in defined uses at specific levels. Any additions or changes to these uses or levels would necessitate a company submitting another application or an amendment prior to us evaluating these new uses. So the GRAS provision has been in effect since 1958, And while the provision itself hasn't changed, FDA's administration of the GRAS provision has. So after the Food Additives Amendment Act was passed, FDA generated what was known as the GRAS list, which were substances that did not need a formal pre-market review because their safety had already been established, either because of a long history of safe use in food, or it was a substance that we had a great deal of information already, and so a review was deemed not necessary. The list was incorporated into the CFR. However, there were many substances that the food industry felt were GRAS that were not included in this list. So in effect, what happened in the 1960s were that many food manufacturers requested that the FDA offer an opinion letter on a substance that was not included in the GRAS list. And these were informal letters that were generally only available to the requester. But this process was officially revoked in 1970 when President Nixon directed the FDA to evaluate the safety of GRAS food substances. This review was not carried out by the FDA, but instead was contracted to the Federation of American Societies for Experimental Biology, known as FASEB, and they selected scientists to make up the Select Committee on GRAS Substances. And this SCOGS committee, as it was known, reviewed and evaluated each GRAS substance. This occurred from the 1970s through the early 80s, and they reviewed over 400 GRAS substances during that time. At the same time, the FDA established a process known as the GRAS affirmation petition process, where the FDA could affirm the GRAS status of substances. So after the SCOGS review of a substance, FDA would review the SCOGS opinion, and then they would issue a final rule in the CFR affirming GRAS status. This petition process continued, with food manufacturers being able to submit these GRAS affirmation petitions up until 1997, at which time the FDA proposed a notification procedure where the food industry would submit a GRAS notification and FDA would evaluate whether there was sufficient information in the notice to support a GRAS determination. This would result in letters that were sent to the notifier. And this is the current program. The final rule was passed in 2016. So let's delve a little bit deeper into what GRAS is. In the provision, it stipulates that there are two different paths to achieve GRAS status. The first and much more common route is through scientific procedures. The company or notifier can establish GRAS status by referencing the available scientific literature and knowledge on their substance. Importantly, I would say that the quality and quantity of data is expected to be the same, whether you're submitting through GRAS or you're submitting through a food additive petition route. So if you don't use scientific procedures, the other route to GRAS is through history of common use in food prior to 1958. Now, you can imagine that this path is becoming increasingly more rare to be used. And in my tenure at the FDA, I have yet to actually see a GRAS notice submitted based on history of common use prior to 1958. So now that we've established the two pathways to get to GRAS status, let's talk about what makes GRAS a bit unique compared to food additive petitions. The GRAS provision stipulates that the scientific data used to arrive at a GRAS conclusion must be both generally available and generally accepted. So in effect, this means that the evidence to support your GRAS conclusion must be published and must be available to the general public. So this could be through a peer-reviewed journal article. It could be through a textbook, for example. Data cannot be confidential, and this is in direct contrast to a food additive petition. In a food additive petition, the manufacturer can submit confidential study that they've generated that is not published, and this would be acceptable for FDA evaluation. But we cannot do this in the GRAS program as it does not satisfy the requirements of general availability. Additionally, the provision states that there should be a general consensus among scientific experts regarding safety. And so again, it helps to have your safety data published in a peer review journal, as this helps to establish general acceptance. So we've discussed the two paths to GRAS and the requirements for the data that are included in the submission. So this slide details what else is included in our GRAS notices. And quite frankly, it's very similar to what we would see in a food additive petition. Companies will provide information on the identity, the manufacturing specifications, dietary exposure. They'll tell us which path they're using, either common use before 1958 or through scientific procedures. And then they'll offer a safety narrative with generally available and accepted data that helps support their conclusion that their food ingredient is safe. So we've talked about the safety standard for the food additive petition, and that is reasonable certainty of no harm. And in the case of the GRAS provision, we have that same exact safety standard. So not surprisingly, the review teams for GRAS notices look very similar to the review teams for food additive petitions. We also have a multidisciplinary team comprised of a chemist, a toxicologist, and where needed, we will include a microbiologist, a nutritionist, occasionally. So in the example of we are reviewing an ingredient that may be added to infant formula or to a USDA regulated product, such as meat, we will work with our colleagues in the infant formula and medical food science team or the USDA Food Safety and Inspection Service. And just like a food additive petition, this whole process is managed and coordinated by a regulatory review scientist. So in the food additive petition, the final result is a regulation for the petition substance in accordance with its intended uses. In contrast, the GRAS notification program, we don't have a regulation, and instead the notifier will receive a letter. There are 3 different types of letters that they could receive. The first and the desired result is what we call a no questions letter, where the FDA does not question the notifier's GRAS conclusion. And most notifications go this route. Occasionally, we will have an example of a notice where we do not believe that the notifier has provided sufficient evidence, and we will reach out to them and ask them to cease to evaluate this notice. And we'll provide a rationale for why we would like them to cease, ask for a cease to evaluate. And in many instances, the companies that receive this letter will end up resubmitting their notice once they either obtain additional data to provide or improve the quality of their submission. Finally, very rarely we'll run into situations where a company will disagree and they will not ask us to cease to evaluate. And when we find ourselves in that situation, we will issue what is called an insufficient basis letter. This is probably the worst of the 3 letters that a company could get. So a no questions letter, it is helpful for the company when they go to try to market their substances, as most manufacturers today really expect that these companies will come with a no questions letter. And in effect, what this means is that while the GRAS notification program is voluntary, most companies will still want to submit their notification because that no questions letter is quite valuable to them. So as I've said, GRAS notifications do not result in formal rulemaking. And so to ensure complete transparency, all GRAS notices are actually available online. And so this includes the GRAS notice itself, which you can access. You could also access our letter, our final result, whether that's a no questions letter or a cease to evaluate letter. And so this means the general public has complete access to all the data that was used to help determine that safety conclusion. So here I just want to summarize the key similarities and differences between the two regulatory pathways that I've discussed. The key similarity is the safety standard of reasonable certainty of no harm is shared by both the food additive petition and the GRAS notification program. However, in the case of food additive petitions, these substances are ultimately evaluated by the FDA, and we own that safety conclusion. A successful petition results in a regulation in the CFR. This is in direct contrast to the GRAS notification program, where the GRAS conclusion is ultimately the company's conclusion. So the program is voluntary. However, unlike the food additive pathway, data for a GRAS notification must be published, it must be available to the public, and there needs to be a consensus in the scientific community that the ingredient is safe for its intended uses. So now that I've outlined these two main regulatory pathways for food ingredients, I want to delve into a little bit more detail about the actual safety assessment before we pull all this together with a case study. And I'll point out that as the safety standard is the same for both the food additive petition and a GRAS notification, this overview of the safety assessment will apply for both pathways. So when approaching the safety assessment, the toxicologist is trying to decide if the data support a conclusion that there is a reasonable certainty of no harm to the consumer. And because we're focusing on consumption under specified uses, remember, it's intended use of the substance and not the actual substance itself that we're reviewing. We rely heavily on the chemist on the team to assess information on the identity of the ingredient. So for example, in the manufacturing process of this ingredient, are there contaminants? And at what level are these contaminants at? These are questions the toxicologist will ask. We also need the chemist to confirm how much of this substance a consumer will eat or drink. And from that, the toxicologist will take that information and they'll look at the known hazards or risks for that substance and at what levels those hazards or risks are observed, and we'll determine what is considered a safe level of consumption. Now, hopefully that exposure does not exceed that safe level. And something else that often surprises people is that when we do our safety assessment for food ingredients, we are actually not performing a risk-benefit analysis. In fact, we don't look at benefits at all when we are considering these substances. This is in contrast to what happens with, say, a drug safety assessment, where typically you are weighing the risks and the benefits. We don't do this because with food ingredients, unlike drugs, drugs are typically not consumed for the entirety of one's lifetime, but food and beverages are. So in many ways, a food ingredient safety assessment is actually more strict because we only rely on safety data and we don't take into account any sort of benefit analysis. So I alluded to the basic elements of a safety assessment on the previous slide. We want to know what the substance is, are there impurities, are there contaminants? We want to know why it's being added to food. And which food it's going to be added to and at what levels. That information allows us to calculate the exposure estimate to determine how much people will be consuming. And from there, the toxicologist can assess the known safety data and determine whether the dietary exposure is too high. So to get a little bit more technical about exposure, we have terminology that chemists and toxicologists use to refer to these numbers. The first is EDI, or estimated daily intake, and this is the amount of a substance that a person is expected to consume in a day, and it's typically calculated from food categories and maximum use levels that are supplied in the petition or notification. And an EDI can be calculated for specific age ranges, like 2 to 5, 18 plus, or it could be calculated for the general population. We call this a 2+ calculation. In contrast, the ADI, or acceptable daily intake, this is the amount of a substance that can be consumed on a daily basis that we would expect no harm to the consumer. So how do we calculate an ADI? Well, it's derived from toxicology studies, which are often carried out in animals. And in these tox studies, something called the NOEL, or the No Observed Adverse Effect Level, is established. And just as its name implies, in these studies, there are a range of doses given to animals for various lengths of time, and we'll look at the highest dose where there's no adverse events being reported, and that becomes the NOEL. From there, you can calculate the ADI by using the NOEL and applying uncertainty factors to account for various things. So for example, most of these studies are carried out in rodents, but this is going to be consumed in humans. So we have interspecies variability that we need to account for. These uncertainty factors, they help to build in a buffer to further ensure safety to the consumer. Ultimately, the toxicologist is going to be comparing the ADI to the EDI, and we hope to see an EDI that is lower than an ADI to ensure safety. So to pull this all together, the safety assessment is done by a multidisciplinary team, and we rely on each other to address different parts of the application to arrive at the final safety conclusion. So you have the chemist who's responsible for what's in this blue box here, the identity and exposure assessment. And then you have the toxicologist who's responsible for the toxicity data outlined here in this green box. And I'm going to go over a few of these studies that we see because you'll see this in our case study. Often we'll review acute or chronic toxicity studies. Typically, we will also look at something we call ADME, or absorption, distribution, metabolism, and excretion data. We will also evaluate genotoxicity data that tells us about the potential for DNA damage from a substance. Very often, we also have access to what are called DART studies, or developmental and reproductive toxicology studies, and human tolerance studies, which will tell us about the potential for GI upset or allergenicity, for example. And finally, the toxicologist understands that there is uncertainty in risk assessments. And so we will incorporate these uncertainty factors. I've already talked about interspecies variability, so accounting for differences between rats, mice, and humans, for example. We also have intraspecies variability. So humans have a tremendous amount of variability due to differences in age, differences, you've got healthy populations versus sick populations, and you also just have a tremendous amount of genetic variability that you need to account for. And as toxicologists, we can also look at the studies and apply a safety factor if we feel that there were deficiencies in the study design. So now for the final part of the talk, let's take what I've told you about the regulatory review process for food ingredients and the general approach to a safety assessment, and we'll apply that to the safety assessment that was done for Steviol glycosides. So non-sugar sweeteners, these are, well, they go by many names. They can be called just sweeteners, sugar substitutes, low and no calorie sweeteners. They're all a class of ingredient that are used to sweeten or enhance the flavor of food and beverages. And as you can see from the chart, they are oftentimes much sweeter than table sugar, meaning that less of it can be added to achieve the same level of sweetness in a food or beverage. So what are Steviol glycosides? Well, the Stevia rebaudiana plant is native to South America, and Steviol glycosides are components of the stevia leaves. They all have a basic common backbone called steviol, and they include compounds such as stevioside and rebaudiosides. The most abundant Steviol glycosides extracted from stevia leaves are stevioside and rhabdioxide A. As a class, they are about 200 to 400 times sweeter than table sugar, but there is some variation there. So, for example, rhabdioxide M is a lot more bitter than some of the other rhabdioxides. And finally, Steviol glycosides are currently used as a general purpose sweetener, and they can be found in a variety of foods and beverages. So Steviol glycosides have an interesting regulatory history. Between the 1970s and the 1990s, FDA received petitions as well as GRAS affirmation petitions for stevia leaves as well as stevioside, but none were formally reviewed, and this was due to deficiencies in the safety data. And in fact, in 1991, FDA issued what we call an import alert, banning the importation of stevia in all forms due to the inadequate toxicology data demonstrating the safety of stevia. But in 1994, Congress passed what was called the Dietary Supplement Health and Education Act. And this resulted in FDA modifying the import alert in 1995 to allow Steviol glycoside products into the US, provided that they explicitly stated that they were labeled as a dietary supplement. So you still could not import Steviol glycosides for food ingredients. 13 years later, in 2008, the FDA completed its first evaluation of a stevia-related GRAS notice for rhabdioxide A. So we have seen quite a few GRAS notices now for Steviol glycosides, and we typically see GRAS notices for one of three types of sources of Steviol glycosides. The first are highly purified extracts from the leaves of the stevia plant. So for example, we've reviewed rhabdioxide A purified from the leaves of the stevia plant. We also see Steviol glycosides that have derived by enzyme modification. And then finally, because the processing and purification from the stevia plant leaves is rather intensive, more and more food manufacturers are turning to microbial fermentation as a way to produce Steviol glycoside. And to date, the FDA has evaluated over 70 GRAS notifications. So I wanted to remind you of this slide that I showed earlier regarding the safety assessment for food ingredients. And in this case study, I'm mostly going to focus on toxicity data, this green section here, that has been used to establish safety for Steviol glycosides. So in discussing the regulatory history of Steviol glycosides, I mentioned that we have an import alert, which was first put into place in 1991. And while it's been modified over the years, it's still in place for any Steviol glycoside preparation that is less than 95% pure. And so why do we have this import alert? I hinted at the fact that it's due to inadequate toxicological data. So to be more specific, we actually do have studies, toxicology studies done with crude or partially purified stevia extracts. And they've unfortunately reported a number of adverse effects, including decreased fertility, as well as renal and cardiovascular effects, such as increased urine output, sodium excretion, and lower blood pressure. And so in response, scientists decided to undertake rather extensive toxicological studies with the hope that more pure preparations of Steviol glycosides may result in fewer adverse effects. And so I'm going to present over the course of the next few slides some of this tox data that has resulted in successful GRAS notifications for specifically purified Steviol glycosides. So some of the critical primary data that we look for when we're assessing safety of food ingredients is something we refer to as ADME data. which stands for absorption, distribution, metabolism, and excretion. Basically, what this means is what happens to stevia glycosides after they're consumed. So we're fortunate to have a rather extensive set of published in vitro and in vivo ADME studies done in multiple species, rats, mice, pigs, and humans. And it's important to note that all stevia glycosides share the same chemical structure. So it's expected that they would undergo the same metabolic fate following ingestion. And from these studies, we know that stevia glycosides aren't broken down by digestive enzymes or absorbed into the upper GI tract. Instead, they are degraded by the gut microbiota in the colon. And this results in the release of Steviol. Steviol is quickly absorbed and conjugated to a metabolite, Steviol glucuronide, in the liver. And in humans, Steviol glucuronide is ultimately excreted in the urine, and any Steviol that is unabsorbed is excreted in the feces. So in addition to a wealth of ADME data, we also have a number of acute and subchronic toxicity studies with stevia glycosides. I'm highlighting 4 of the published studies on this slide. There are more than this, but these are 4 commonly cited ones. They range from 28 days up to 90 days with doses as high as 4.5 grams of stevia glycosides per kilogram of body weight per day were given to rats. And these typically have been conducted in rats, and the stevia glycosides given are the most abundant ones, usually stevioside and rebaudioside A. And as noted, there were no signs of toxicity or treatment-related findings associated with these studies, but occasionally they did note decreased weight gain. But this would not be unexpected given that stevia glycosides have a lower nutritive value. In addition to the short-term toxicity studies, we also have a number of in vivo and in vitro genotoxicity assays that have been published. These assays are designed to detect DNA damage, and I've summarized several of them here in this table. As you can see, there are a variety of different assays have been performed, and they've demonstrated that stevia glycosides are not genotoxic. There are 1 or 2 publications indicating that particularly stevioside may be genotoxic. There was a group that published in 2007, it's Nunez et al. However, I'd point out with this study, they did not use proper controls. And they also used a COMET assay, which detected damage 5 weeks after administration, and typically COMET assays detect short-lived genetic damage. And so oftentimes we have to take a weight of evidence approach, and the overwhelming majority of the studies show that there are no genotoxicity concerns with stevia glycosides. In addition to those genotoxicity studies, we also have chronic toxicity studies and a 2-year carcinogenicity study done with various doses of stevia glycosides as high as 2 grams per kilogram body weight per day. And these have all been published. Importantly, there are no increases in cancer incidence compared to control animals in any of these long-term studies. So if you remember from my introduction on stevia glycosides, we do have an import alert on stevia glycosides that are less than 95% pure. And the major reason for this were some noted adverse effects with crude extracts, and this included decreases in male and female fertility. And we had evidence as early as 1981 that if you used a more purified extract, that these treatment-associated effects on fertility were not observed. But it was really some additional developmental and reproductive toxicity studies that were done in 1995, another one done later in 2008, that helped bring consensus to the scientific community that consumption of purified stevioglycosides does not induce changes in male or female fertility, and it also does not result in developmental adverse effects. So with the exception of some of the ADME studies I first presented, most of the studies on stevioglycosides that I've talked about so far have been done in animal models, but we're pretty fortunate to also have a good amount of human clinical study data with stevioglycosides. I can't possibly go through every human clinical trial that we have published at this time in the short amount of time I have today, but I've summarized 3 of the most frequently cited studies that were done really to address some of those initial toxicity concerns from the crude extracts related to cardiovascular and renal effects. So these published studies were done in both healthy adults as well as patients with type 2 diabetes. Study subjects consumed 1 to 1.5 grams of stevioglycosides per day for up to 4 months. And as indicated on the slide, there were no significant changes in parameters such as blood pressure, glucose levels, HbA1c levels, or insulin levels. So to summarize, there was some initial concern with stevioglycosides that were either crude or partially purified due to adverse effects on reproductive fertility and cardiovascular effects that were noted in those early studies. But later studies done with purified extracts of stevioglycosides have shown very little signs of toxicity, genotoxicity, carcinogenicity, no developmental or reproductive effects, and the data has been very encouraging in human studies as well. So we've talked about the toxicology studies used to establish safety, and now I want to get into the exposure estimates a little bit. So there's actually a publication that we use done by Renwick et al. From 2008, and it presents a method to calculate dietary exposure for sweeteners. And their method is based on published consumption data, and it takes into account that sweeteners are inherently hundreds of times sweeter than sucrose. So you need lower amounts of your sweetener to be added to foods to achieve that same level of sweetness. And a critical assumption that they use is that a sweetener would replace the use of either sugar or another sweetener in the food. And so additionally, oftentimes our submissions are for one particular stevioglycoside, such as ribodioside A, and we need to compare that to compare the EDI from that ribodioside A to the ADI from the tox studies. And many of the tox studies, they were done with stevioside. So we have to be able to normalize between ribodioside A, ribodioside M, for example, to stevioside. And fortunately, we can do this because all of these stevioglycosides share the same common steviol backbone. So what we can do is actually convert to steviol equivalents. And I've got an example here on this slide of how we do that. It's a ratio based on molecular weight. So you can see the molecular weight of steviol is 318 grams per mole. Stevioside is 805 grams per mole. So we take a ratio of that as 0.4, but we can do that for, we can calculate that ratio of steviol to ribodioside M to ribodioside A, and we have the different ratios. So I wanted to quickly walk through an example of comparing that ADI to the EDI. So the ADI, as I said, is derived from the toxicology studies. And we typically use the longest toxicology study to cover any potential adverse effects that could come up. And what we have, our longest study we have for stevioglycosides is a 2-year rat carcinogenicity study. This was done with stevioside. where the NOEL was the highest dose tested, 970 milligrams per kilogram per day. We normalize this to Steviol equivalents, which is the calculation I showed you on the prior page. So our normalized NOEL becomes 388 milligrams per kilogram per day. And then we apply the safety factors to account for different inter and intra-species differences. Traditionally, this is a factor of 10 for each of those, for a total of 100. And so after applying that uncertainty factor, we arrive at an ADI of approximately 4 milligrams per kilogram per day. So that's our ADI, and now we need to compare it to our EDI. And I just pulled an example from a recent GRN or GRAS notice for ribodioside M, which was as a general purpose sweetener in foods. The first column here is taken directly from the Renwick paper, and it shows that the intake of sucrose for adults and children on a body weight basis. Then the next column, we would divide by the relative sweetness of ribodioside M. So since ribodioside M is 200 times as sweet as sucrose, we would need 200-fold less in an ingredient. So we would divide 675 by 200 to get a level of 3.38 milligrams per kilogram per day. And then in order to compare to the ADI, we need to convert this to Steviol equivalents. So the conversion factor for ribodioside M is 0.25, and so we end up with an EDI of either 0.85 milligrams per kilogram per day in adults or 1.24 milligrams per kilogram per day in children. And fortunately, this is lower than our ADI of 4 mg per kilogram per day. So I wanted to finish up with just noting that globally, there are over 60 countries that have approved Steviol glycosides as safe. And notably, this includes Europe, where the European Food Safety Authority, or EFSA, has also reviewed the safety of Steviol glycosides and has approved their use as a sweetener in foods and beverages throughout the EU. And so this is my last summary slide, and I just want to summarize the main points from today's talk. First, the FD&C Act gives the FDA the authority to oversee the safety of the food supply. In 1958, amendments to the FD&C Act resulted in the definition of a food additive, as well as the GRAS provision. The safety standard for all food ingredients, whether submitted as a food additive petition or GRAS notification, is the same, and that is reasonable certainty of no harm. And importantly, it is the intended use, but not the food ingredient itself, that is assessed for safety. And finally, food ingredient assessment is based solely on safety. We do not perform a risk-benefit analysis. And so that is everything, and I would be happy to take questions at this time. On this last slide, I have a few websites for reference that may be helpful if people would like some more information on sweeteners, on the GRAS program. If you'd like to peruse our GRAS inventory, that site is there, as well as more information about our food additive petition process.
Okay, great. Thank you very much for that presentation. It was very comprehensive. We do have a number of questions that have come in. So I will leave this slide up so our attendees can screenshot it or have this information as a reference as we move forward. So I think you went through a very comprehensive example about how levels are assessed, reasonable amounts to consume and high consumers. Where does the intake data come from?
So there are multiple different ways that the company or the or FDA can get data. Most commonly, we have something called the NHANES survey, which are surveys that are done every few years of the American public. And they're dietary surveys, so people will recall what they've consumed over the course of 1 day, 3 day, 5 day blocks of time, and that data is compiled, and then food categories are assigned, and our chemists can look at different food categories and how many data points they have for each, and calculate use
From what we eat in America
based on that
survey.
survey. So that survey is done every few years to update it to changing consumer habits.
Okay, and then you also mentioned the intended use of these ingredients. The ingredient itself isn't approved, it's the intended use. So how does FDA consider combinations of sweeteners? When more than one sweetener is used, does that necessitate a new GRAS or food additive petition application?
So that's interesting. FDA does not consider mixtures at all when we are looking at food ingredients. So we don't, a GRAS or a petition, it's going to be submitted for an individual sweetener. And so we don't consider the fact that it gets mixed in with other sweeteners, for example. This is not part of the review process.
So as long as the intended use is the same, once a sweetener has been approved for that use, then it could be combined with another sweetener in a food formulation.
It can, yes.
Okay, and then there was a question about, can you give an example of when a safety status could be revoked and why?
So, for example, I mean, I would say that this is not something that happens very commonly. However, if there were an overabundant amount of information that would suggest that something is no longer safe, that would likely first result in it being paused from the not being added into the food supply while we would do a new safety assessment. Another example is if we had evidence to suggest that consumption was much higher than what the initial dietary exposure estimates that were provided in the notification or petition. If we had evidence to believe that exposure was much higher, we could pause that ingredient in the food supply until we could adequately reassess that exposure. Those are two examples that come to mind of reasons why we would reassess, but I can't come up with any specific examples of times that has actually happened, but it is an avenue that we can take if we feel we have the need to do so.
So that's a perfect lead into what I think is our last question, and that is certainly the food supply is continually changing, and new non-nutritive sweeteners continue to enter the marketplace as they are reviewed and approved. So is there a period of time, is there a time frame by which FDA periodically reviews intake assessments as the food supply changes, or does there have to be like a precipitating event for that re-review of intake assessments to occur?
So intake assessments will be reviewed anytime a substance comes in. And so ultimately, what I mean by that is that if somebody submits a sweetener GRAS notice, for example, and then 6 months later we get another GRAS notice for that same sweetener, I can use stevia, for example, we are routinely redoing those dietary exposure estimates every time a submission comes in. And so I mentioned with Steviol glycosides, we've reviewed 70 applications, over 70 applications since since that first one in 2008. We routinely have 3 or 4, at the very least, Steviol applications each year. So we are continually looking at those dietary exposure estimates anytime we get a new application or an amendment to an application for a sweetener or for any ingredient.
Well, that's great to hear. That is all the time we have today. So I want to thank you, Dr. McGregor-Das, for your wonderful presentation. I also want to remind those in attendance to complete your post-webinar survey to complete your participation in the study. And if you have any questions about the study, this webinar, or the Institute for the Advancement of Food and Nutrition Science, you can send me an email at tzek@ifns.org. You can also find information on our website and by connecting with us on LinkedIn. Thank you all for your participation, and I appreciate your time today. Take care.